Hi students, here we are going to discuss about the double helical structure of DNA. It is put forward by Watson and Crick. So it is known as Watson-Crick model. So DNA is the deoxyribonucleic acid. So the sugar unit that is present in DNA is a 2-deoxy-D-ribose. And the nitrogenous bases are present in DNA are adenine, guanine, cytosine and thymine. And DNA consists of two strands of a polynucleotide chain that are coiled around each other and known as a double helical structure. And nucleotide making up each strand of DNA are these. Each strand is means one polynucleotide strand is connected through this phosphodiester bond that is already seen in RNA. The phosphodiester bond is formed between C5 dash of one deoxyribose moiety to the C3 dash of the adjacent one, and this forms the backbone of these DNA strands. So this double helical structure it is formed. So one strand it contains two strands of polynucleotide. This one strand is each strand is linked through by means sugar unit that is present in one strand is linked through phosphodiester bond, likewise that in another strand. So the C1 of this sugar unit is pointing towards the axis of helix, pointing extended inward towards the axis of helix. So this C1 carbon, that is first carbon of this each sugar moiety is connected through the base unit. You can see that this carbon is connected to the base unit. These are the one strand of polynucleotide, that DNA, and this C1 is extend towards inward the helical axis. Then so we can say that sugar phosphate backbone forms a periphery of the spiral structure, but the base are on the inside of this helix. And one thing we have to note that two strand is present there, but these two chains are runs in an opposite direction. That means these sugar units are oriented in opposite way. C3 dash is to C5 dash in one chain, then another one will be that C5 is to C3 in the other chain. Base in one strand of DNA are paired with the base of other strand of DNA. DNA contain two polynucleate chain, two strands. Okay, so one polynucleate chain contained base is paired with that second strand base, that is paired by hydrogen bonding, that is linked through by hydrogen bonding. That hydrogen bonding is between this base. That base pair will be adenine with thymine and guanine with cytosine. So this adenine and thymine will form through two hydrogen bond that you can see here. This is the adenine and this is the thymine. So adenine and thymine is bound together. These are the hydrogen bond that is present in the adenine and thymine base pair. So here only two hydrogen bonding is formed, but in the case of guanine and cytosine there are three hydrogen bond is present. So this hydrogen bonding is cytosine will bonded with the guanine only, thymine will bonded with the adenine only. That it will not allow the cytosine with adenine or thymine with guanine. So as a result, what will happen? Combination of another means DNA, right? Same like sequence will not occur in the two type of strand. So sterically and energetically formed only AT and GC pairing in DNA. This is known as base pairing principle or base complementarity. Two strands of DNA are not identical, but we can say that it is complementary to each other. That sequence is one strand is automatically determine that of the other, because one sequence will be in one strand, that sequence reverse order will be in another strand. So one strand sequence will determine the another strand's sequence. The portion of one strand of DNA had the sequence of example. We are telling the example for this. When one strand of this DNA is sequence is adenine, thymine, guanine, cytosine, thymine, cytosine, adenine, then it's the complementary will be the another one. Corresponding complementary strand would be. So adenine means its complementary will be thymine. So next is thymine, complementary is adenine. Guanine's complementary is cytosine. Likewise, it will be. The double helical secondary structure of DNA thus resembles a spiral staircase structure. Means it is the secondary structure of DNA. The secondary structure of DNA thus resembles the spiral staircase like a structure. This sugar phosphate backbone of two strands forming railing. Okay? Staircase ആണെങ്കിൽ അതിന്റെ railing will be എന്താണ്, that will be the sugar phosphate backbone, and that step will be the base pairing, that nitrogen bond base pair. And the DNA molecule contain equal number of adenine and thymine unit and equal number of guanine and cytosine unit. A molecule is about to 20 angstrom in diameter. The base lie in a plane that are approximately perpendicular to the helix axis. Base pair will be perpendicular to the helix axis, and the average distance between two adjacent pair, base pair, one strand to another strand will be, the average distance is 3.4 angstrom. The complete turn of helix occurs. One helix turn occurs every 34 angstrom unit. So this 34 angstrom unit, one helical unit turn contain 10 base pair per turn in a double helix structure. That means you know that helical structures, one turn means, and see here, one turn will be this, from here to this is the one turn. That one turn contain, that one turn distance is 34 angstrom. So that one turn contain 10 this base pair. So specific sequence of the base in polynucleotide chain is very important. That is specific sequence of base is known as primary structure, and it carries genetic information. And these are the structure of double helical structure of DNA. Next video we are going to discuss about the biological function of these nucleic acids. Biological functions are to replication and biosynthesis of protein, that we can see in the next video. Thank you.